So the presentation, as Jen said, it's on lab diagnosis of diphtheria. But just first of all, just to give you an overview of the disease itself, diphtheria. The main causative pathogens, or the main pathogen is of course Corynebacterium diphtheriae. And there are of course other pathogens that can potentially cause the disease, the clinical disease diphtheria. That is C. ulcerans and C. pseudotuberculosis. The latter two are zoonoses, and C. pseudotuberculosis is in fact quite rare in causing the disease. The incubation period of the disease is approximately 2 to 5 days. It can be longer, it can be up to 10 days. The distribution of the organism is worldwide, but it's usually quite uncommon in countries that have good and effective childhood immunization programs. The primary source of the organism is the upper respiratory tract, the nose or throat. And in places like in the tropics, where skin diphtheria is particularly common, obviously infected skin lesions. And C. ulcerans, as I said, is a zoonotic pathogen and can be found in many animals, and particularly companion animals such as cats and dogs. Pseudotuberculosis is more sort of linked with goats and sheep. Spread is normally person-to-person contact. It's respiratory transmission. The other zoonoses, C. ulcerans in particular, can be milk-borne. And as I said, these organisms may be found in domestic pets. And as we all know, vaccination is the most effective tool in preventing the manifestation of clinical disease caused by all three toxin-producing species. And I have to say, it's the toxin that is the main pathogenic factor within these organisms. So these are organisms that produce diphtheria toxin. There are two main types. As I said, respiratory disease is the most common, and cutaneous diphtheria. And the symptoms of respiratory diphtheria can be quite mild, mild pharyngitis, to very severe disease with the formation of the classic pseudomembrane. If untreated, it can lead to very severe complications of the heart, myocarditis, of the nervous system, and of the blood. And it can also affect the neurological system. As I said earlier, the case fatality rate is quite high, and even in the younger population, it can be as high as 20%. Fortunately, there is treatment, that is diphtheria antitoxin, and of course antibiotics, usually penicillin or macrolides, such as erythromycin. I'll just show you some clinical pictures. This is a slide showing the classic pseudomembrane. This is a very thick, very rigid membrane which forms at the back of the throat. It's very, very difficult to remove the membrane. If you remove it, it may cause excessive bleeding. So it needs to be dealt with and removed by an expert, by an infectious disease expert. And just to show you, this is a membrane from a case of C. ulcerans that was removed from somebody's throat. And with diphtheria, you tend to also present with what is called the classic bull neck syndrome, and that is the inflammation of the lymphatic lymph glands, and this causes this very, very severe presentation. If a membrane is present, the patient may have difficulty, or usually does have difficulty in breathing, and therefore a tracheostomy has to be performed. This is rather a very messy tracheostomy, and unfortunately this is a case, this is a terminal case of diphtheria where excessive hemorrhaging occurs in many of the body orifices, particularly from the nose and the mouth and the ear. This is showing cutaneous diphtheria. These are very classic lesions. They're very difficult to treat. These particular lesions can last for days or even months. Okay, so what is the clinical definition of a case of diphtheria? So these are the case definitions. The suspected case is an illness of the upper respiratory tract characterized by the following. So pharyngitis, nasopharyngitis, and the presence of this pseudomembrane. The final classification or the final confirmed case is based upon the isolation of a toxigenic strain of C. diphtheriae or C. albicans. Okay, fortunately, as I said, diphtheria can be prevented, vaccination being the main weapon, if you like, and also very rapid and prompt clinical recognition is extremely important. So if a case is identified, treatment is normally initiated with diphtheria antitoxin and also antibiotics. The other very effective and very important tool is, of course, good surveillance, both epidemiological and microbiological. Okay, so diphtheria antitoxin, this is, I would say, a very, very old therapy, and it used to be made almost everywhere, but now it's unfortunately down to very few countries in the world. But it is extremely important for each country to have a stockpile. We really need to have, in fact, a global stockpile. As I said, the diphtheria antitoxin was first discovered in the early 1900s, and the technology of producing the antitoxin hasn't really changed very much. It's still made by injecting horses with attenuated diphtheria. But more recently, over the last couple of years, researchers in Braunschweig in Germany are trying to change that. They've been working on developing a recombinant antibody molecule, kind of building it genetically in the lab and amplifying it through cloning rather than infecting animals. And this has been extremely successful. It's still in development and hopefully may be commercialized quite soon. So I think this is a real revolution in the in therapy for diphtheria. This is the toxoid from the 1930s. And this is the gentleman who discovered diphtheria toxin and also tetanus toxin, Emil von Behring, and he won a Nobel Prize in 1901 for his work using animal antibodies to create these medicines. And obviously there was very wide promotion of diphtheria in the 1930s. This is a campaign or a poster from a campaign in the US. Okay, so surveillance. Why is surveillance so important for diphtheria? Okay, it's obvious, I think. It's to understand disease epidemiology, to monitor the disease trends and also the distribution of strains over time, to inform, to identify outbreaks rapidly and in a timely manner, and also to inform response. It's important to evaluate how effective the vaccine program is, so as to inform public health planning and vaccination policies. However, I have to emphasize, again, very important for microbiological and epidemiological surveillance. And microbiologists and epidemiologists must work together for surveillance to be efficient. This is a publication which is available from WHO. It's the Vaccine Preventable Diseases Surveillance Standards, which provides a lot of information on surveillance. There's a chapter on diphtheria. It gives you the reasons for surveillance and, very importantly, the evaluation and also steps of an outbreak investigation. So there are disease-specific chapters. The web link is at the bottom of the slide. And these are the VPDs that are included in the surveillance standards. So it's primarily 22 diseases. And it describes, for example, case investigation. How do you undertake case investigation? So basically, one lab confirmed case should trigger a public health response immediately. If you have two geographically linked cases, then that is an outbreak. And so every single case should be investigated. So basically, two samples are collected, usually samples of the upper respiratory tract. Case has to be isolated, has to be treated with antitoxin and antibiotics, immunized with diphtheria toxoid, and contact tracing has to be undertaken. So in an outbreak response, in a highly vaccinated population, we normally follow the procedure that I just described. You identify contacts, you monitor the contacts, you collect specimens, give antibiotics, and vaccinate if the vaccination is not up to date. In a poorly vaccinated population, again, contact tracing is still very important. Antibiotics, absolutely life-saving. And mass immunization should be considered, which is based on the epidemiology of the disease. I'd just like to show you some data slides from WHO. This shows the number of cases reported to WHO between 1980 to 2019. This is, covers approximately a 40-year period. And if we look at, in particular, this purple line, this is the European region. And this, as many of you might know, refers to the very devastating outbreaks that occurred in the Russian Federation in the 1990s. This was obviously due to the breakdown of the former Soviet Union and the obvious breakdown of vaccination programs. During the peak of the epidemic, more than 50,000 cases were identified. The other area that I will draw your attention to is this, this blue line, which is the Southeast Asia region, where we're seeing quite a few outbreaks within this region. And likewise, this orange line, which is the Africa region. So these are regions, if you like, of concern. So despite all the progress, preventing and treating the disease, diphtheria has not been eradicated, and it still flares up all over the world. And outbreaks tend to occur in places that have been destabilized by population migration, civil unrest, and so on. So places like Bangladesh, Yemen, Myanmar, and so on. This is the number of cases over the last 10 years in the WHO European region. So we are still seeing cases, although sporadic, within the Euro region. Most of these tend to be imported. If we look at the deaths, as I said earlier, the case fatality rate varies. It's a lot higher amongst children, but on average, the case fatality rate in the Euro region is about 3%. We rarely see cases of death, but we have had them. There were several cases during 2015 and 2016, and more recently, we had cases in Greece and Bosnia, and these were primarily refugees from Afghanistan, where the disease is unfortunately endemic. And one of the most sad and devastating areas is, of course, Yemen. There were thousands of cases, and they're still suffering from not only diphtheria, but other infectious diseases, in particular cholera. And just to give you an idea, in one particular year, there were more than 1 million cases of cholera and over 1,000 cases of diphtheria. Okay, so that was just an overview really of the epidemiology, the clinical aspects, and surveillance. So now let's go on to lab diagnosis of diphtheria, which is of course a very important area in confirming a clinical case. So as I said earlier, the main organisms are Cranibacterium diphtheriae, ulcerans, and pseudotuberculosis. All gram-positive pleomorphic rods. There are key tests that are done in labs, classic tests like catalase, enzyme tests, which I will describe, cystinase, pyrazinamidase. There are primarily what we call 4 biovars within the species C. diphtheriae. These are mitis, gravis, belfanti, and intermedius. The major virulence factor is, of course, the diphtheria toxin, and this is the gene encoding the toxin is carried by a family of very closely related bacteriophages called carinophages. So if we start from a suspected clinical case, normally 2 samples are collected from the case, a pharyngeal swab, ideally under the pseudomembrane if possible, and a nasal swab. And if possible, if a piece of the pseudomembrane tissue can be obtained, and this is then placed into saline. The type of swab that we use is usually a cotton tip swab, tipped applicator, and always taken prior to starting antibiotics. If, however, antibiotics have started, you can still collect the swab, but obviously you need to know this when you're evaluating the results. The swab is then placed into a suitable transport medium, Amy's, Stewart's, or you can place the dry swabs in what we call silica gel packs. These are special foil packs containing silica gel. These are quite important when you're taking samples in the field and there's going to be a delay to the lab. However, it's important to try to send the samples to the lab within 24 hours of collection. And ideally, they should arrive there within 48 hours. And contact should also have similar samples taken. It's important to collect the appropriate data, so demographic, name, age, etc., reporting information, clinical symptoms, the laboratory, the vaccine status if known, any epidemiological data, has there been any contact with another case, has there been any travel, has there been any animal contact. And this obviously will lead to the final classification of the case. This is an algorithm for lab diagnosis, which you will find in the new WHO manual on lab diagnosis of diphtheria, which has just been published. I will give you the reference, and I am happy to send anybody who wishes to see the manual a copy. This is an e-copy. So basically, it would be taking swabs from the appropriate sites and placed onto the recommended primary culture media. And the recommended culture media are blood agar, Hoyle's tellurite, and Tinsdale. I will explain further regarding Tinsdale in a moment. Primarily, blood agar and Hoyles tend to be the main primary culture media that are used. Tinsdale is more of a selected medium. And then any suspect colonies, again, purified onto blood after microscopy, various tests performed, biotyping, toxin testing, antimicrobial susceptibility testing, and usually they are referred to our national reference lab here at Colindale for confirmation and toxin testing. So classic biotypes have so-called classic colony morphologies. It's often, I think it's quite difficult actually to subdivide them into biovars based on their colony morphology. Mitis, however, they tend to be slightly hemolytic, whereas gravis are non-hemolytic. So this is a culture of Cranibacterium diphtheriae here and here. And you can see they're quite classic grey colonies, non-hemolytic here. And this is CR strands. Sorry, what are we doing? Yes, so this is CR strands here. So they're quite similar. And again, this is C. diphtheriae on blood agar. And this is the organism on Hoyle's tellurite, forming the very classic black glistening colonies. But unfortunately, other flora will also grow on tellurite. Some streptococci tend to grow on tellurite. This is a gram stain showing the classic pleomorphic gram-positive rods. And there are other screening tests or single tests that can be used to potentially identify toxigenic cyanobacteria. The classic tests are these, so nitrate reduction, urea hydrolysis, catalase, cystinase activity on Tinsdale, pyrosinamidase activity, they're normally pyrosinamidase negative, and carbohydrate fermentation, primarily glucose, sucrose, maltose, glycogen, starch. And this is a table showing the biochemical identification of pathogenic cyanobacteria. So the main screening tests, I would say the important sort of initial culture tests are cystinase, PYZ, and urea. C. diphtheriae, all the potentially toxigenic strains are always cystinase positive and PYZ negative. So these two tests are very important. This is cystinase test on the Tinsdale medium. I think this one's a better one. So basically, they will grow as these black colonies surrounded by a brown halo. So they're very easy to distinguish. Many labs actually use Tinsdale as a primary culture medium. Some labs tend to perform the test on slopes. So this is the cystinase test on a slope. This is cystinase positive, this is cystinase negative. And here we have again positive and negative, so very clear cut. This is the pyrosinamidase test, where all C. dips, C. alstra, and C. pseudotuberculosis are always PYZ negative. Other cyanobacteria are PYZ positive. This is a commercially available test. And these are the classic sugars that some labs, particularly those labs in developing countries, are still using. So nitrate broth, urea, and classic sugars. These are the serum water sugars, his serum water sugars. And this is what they look like when they are positive. So this is glucose, maltose, glycogen positive. which means it could well be a gravis biovar. And of course, we have the API Corani kits that many labs still use. These are very useful and extremely accurate. And these are the typical profiles for gravis, mitis, and ulcerans. And of course, many labs, certainly in the UK and also Europe and the US, now are using MALDI-TOF for identification of pathogens. This is a very useful and very accurate technology. Basically, you're bombarding your sample molecules with a laser light to bring about sample ionization. And the whole run can be performed in 4 hours, usually one isolate in 11 minutes. This is reasonably, I would say 99% accurate in identifying Corynebacteria. So this particular method is reliable to species level, and our experience, basically, we find that MALDI-TOF is reliable for C. diphtheria and C. ulcerans. We don't have much data on C. pseudotuberculosis, but it is the method that we would certainly recommend if possible. This is the manual on laboratory diagnosis of diphtheria and other related infections that was recently published. It's an online publication. So as I said, if you have any problems at all, I can send copies of the manual if you would like to perhaps email me after the talk. And we have standard operating procedures. We have SOPs for the investigation of throat-related specimens and also the identification of Corynebacterium species. However, the key test in confirming a case of diphtheria is the test for the detection of the lethal diphtheria toxin. This is a test which is not usually undertaken in a routine diagnostic lab. It's usually undertaken in a reference lab. And unfortunately, laboratory skills in toxigenicity testing have diminished in many countries because of the rarity of the disease. This is the perception, certainly in Europe. And all UK labs normally refer isolates to our reference lab at Colindale for toxin testing. So I think it's essential for international labs to retain skills in this very specialized area of microbiology. So there are the conventional tests and there are the molecular tests. The classic ELEC test was first described in 1949. It's quite a complex procedure and it takes about 48 hours to perform. There was also an in vivo test in guinea pigs, and again, this took up to 5 days. However, we modified the ELEC tests during the 1990s. This is a much more simpler way of identifying the toxin producer, and it usually takes about 18 hours for a result to appear. We've then gone on to other enzyme immunoassays, and also we developed an immunochromatographic strip test. This was done way back in 2000 with one of the US aid agencies called PATH, again in response to the epidemics in Europe. And this was an excellent test. A result was obtained with between 6 to 16 hours, but unfortunately supplies diminished and nobody was interested. We could not attract any manufacturer to produce this test due to the fact that diphtheria was was not present anywhere at all. That again was the unfortunate perception. So we then went on to molecular testing, PCR. The conventional PCR for detection of the diphtheria toxin gene was developed in 1991. And this led to further developments in technologies and platforms. And this, we have a real-time PCR, which was developed in 2013, and also various multiplex PCRs, which were developed over the years, which I will describe briefly. But first of all, this is the classic ELEC test, quite complex. The medium is usually prepared in-house. It's not a difficult medium to prepare, but unfortunately it's not commercially available. So we have controls, we have positive, negative, and weak positive controls, and each test, each plate will accommodate 2 test strains. So normally the culture is streaked across the plate, like so, and an antitoxin disc is placed in the middle of the plate. This is a form of immunoprecipitation, and if the test strain is positive, it will form an arc of precipitation with the toxin positive control. If negative, the arc will not form. As I said, this is a very, very specialized test, and it's quite difficult to prepare if it's not done on a normal basis. Okay, this is much simpler. This is the modified ELEC test. Again, the same medium, the same ELEC medium. And it's immunoprecipitation. So test strain, again, each plate will accommodate 2 test strains along with 3 controls. And if the test strain is positive, it will form an identity with the positive controls, as shown here. It's very simple to perform, and a result is usually made available within 16 to 18 hours. And this is the test that we use in the laboratory and that many other labs tend to use these days. This is the test itself once again. So we have the 3 controls: the negative control, positive control, weak positive, and the strong positive. And these are the 2 test strains here. As I said, novel diagnostic assays have been or are still being developed over the years. First of all, a real-time PCR assay, not only for toxin detection, but for species confirmation, has been developed, and it's possible to undertake the assay directly from culture and also from clinical specimens. The ICS test, or the lateral flow test, is currently, thankfully, being revived under a new EU Horizon programme. And there's also a new innovation that was developed by colleagues in Cambridge to be used in areas such as Southeast Asia. And this is like using a mobile battery-operated mini PCR unit and oligo detection strips. It's quite novel, really. You have your test sample, you have your mobile unit where the sample undergoes PCR amplification. You add your various developing reagents, and you expose the sample to these strips, and you obtain a result. As I said, it's in very early stages of development, but it's quite an optimistic approach. This is the rapid test, which is not yet available. It was a great test, and we used it in the field in many outbreak situations. It had a very high sensitivity and specificity. Unfortunately, it's no longer available, but it is being revived under an EU Marie Curie fellowship programme. but we are still looking for funding for research to ensure these new methods. And we're also looking for potential manufacturers that are willing to produce this product. This is the test itself. So it can either be performed directly from a plate culture or from a swab. So suspect colonies or the throat swab itself is placed into an enrichment broth, incubated, anything from 3 to 16 hours, and then an ICS test strip is placed into the sample. And after 15 minutes incubation, you have these typical results. So this is the control. And this is a positive here. You can just about see it. This is it in reality. This is much better. So this is the control. This is an uninoculated strip. So this is a positive and this is a negative. Okay, so PCR, it's extremely useful. It's rapid and it will confirm potentially toxigenic species. It will detect the diphtheria toxin gene, so it's a good predictor of toxigenicity, but it will not confirm biological expression of a toxin. For biological expression, it's absolutely important to confirm any positive PCR result with the ELEC test. It can be used on mixed cultures and it can generate a result within 3 hours. And it's extremely useful because it can help in decision-making, in public health decision-making of a suspected case of diphtheria. This is the conventional PCR where we're targeting the A subunit. The toxin itself is an AB toxin gene. The A portion is the biologically active portion of the gene. So this is a very good method. It's still used again in many labs, but it can't differentiate between the species. So we developed at Collindale a real-time PCR for species and toxin gene detection. And this is the method that we tend to use for any potential isolate referrals to the lab. All these methods are in detail in the manual. Another important area is, of course, antimicrobial susceptibility testing. It's important that AST is to be undertaken on all C. diff isolates, irrespective of toxin production, and on all isolates from cases and carriers. Okay. There are currently 2 guidelines available for testing and interpreting results. There's the CLSI guideline, but it doesn't have any data or methods for testing Corynebacterium species by disk diffusion. But instead, it recommends the broth microdilution method as the gold standard. UCAS has developed zone size diameters for disk diffusion testing and also breakpoints for interpreting MICs using, again, either broth microdilution for Corynebacterium species and including C. diphtheriae. These are the CLSI and UCAS recommended antimicrobials and the various interpretations. And we tend to perform our ASTs using the E-test gradient diffusion method, which I'm sure many of you are very familiar. Again, all the methods are in detail in the manual. Okay, I said it's important, but why? Okay, there is increasing, unfortunately, drug resistance amongst Corynebacteria, which are challenges in many countries. And one classic example is clindamycin resistance, which is one of the antimicrobials used in therapy. Therefore, it's highly recommended, particularly if clinically macrolides are to be used instead of penicillin. Okay, let's go on to EQA. Again, as we all know, it's very important to have EQA systems in place for specific pathogens, particularly pathogens that are not commonly identified or found, such as Corynebacterium diphtheriae. So for C. diff, for diphtheriae, we have had distributions, particularly amongst EU member states and also other countries, way back in 2010, 2012 and 2013, under the auspices of ECDC. And with those dispatches, we had 6 freeze-dried simulated throat specimens, which were distributed to each country, and the results were documented, and there are various publications on the results. More recently, in collaboration with colleagues at NEQAS, we have a global EQA where we have distributed to approximately 40 countries. So this is again under the auspices of NEQAS and also WHO. And the distributions were made in July, and the results are currently being analysed. So why is it important for diphtheria diagnostics? I think it's quite obvious. It ensures the reliability, obviously, of the results and the epidemiological assessment of disease prevalence. It's essential to monitor lab performance and the quality of results. And it's also important to have regular training workshops coupled with EQA, as it is really important to maintain expertise within these very specialized areas of microbiology. Just to say a little bit about the workshops that we've been doing as part of our role as a WHO collaborating centre. We've held numerous EU and global workshops in the last 2 decades with quite recent increased activity, and they're usually held under the auspices of WHO, mainly in response to need and disease burden. So we've held workshops all over the world for all the regional offices, for Euro, for the Eastern Mediterranean region, Southeast Asia, and also the Africa region. And more recently, due to the COVID pandemic, we've been holding webinars, which they're not exactly ideal, but it was really the best that we could offer at this point. So we've had seminar kind of webinars for Myanmar, for Greece, for recently the Eastern Mediterranean region. And also recently, colleagues at CDC have organised a webinar for the Pacific area region. The first ever workshop was held way back in 1995, again in response to the diphtheria epidemics. And this was held at Colindale, and followed by numerous workshops in places like Indonesia, Surabaya. Again, more recently, a couple of years ago, we had another workshop for colleagues from the former Soviet Union, in Lebanon for the Eastern Mediterranean region, and this was Manila for the Western Pacific region, and the last workshop pre-COVID was in India for the Southeast Asia region. sorry, no, the last workshop was in fact last year in Abuja in Nigeria. This was not held under the auspices of WHO, but it was held under the auspices of the then PHE Global Health Programme, with Nigeria being one of the countries for capacity building. So this was the last workshop that we held. Okay, so there are still many, many challenges. Diphtheria is an uncommon disease in countries with good and effective immunization programs. And it's difficult to maintain expertise in clinicians, public health officials, microbiologists, etc. So I think the greatest challenges still remain as retaining and developing microbiological skills, increasing clinical awareness, and ensuring good surveillance systems. I think the resurgence of diphtheria in the 1990s and also the present day, it's present in every single region. I think it tells us that we really have to be prepared. There was a goal to eliminate diphtheria by 2000, and it's definitely not been met, and a new target still needs to be set. I'd like to just finish by describing this. This is a publication that was brought to our attention from colleagues at CDC. It's a very interesting article in the Smithsonian magazine. And it's kind of like a history, a story of diphtheria. So how science conquered diphtheria, and they called it the plague amongst children. And they said it was highly contagious, lethal, and mysterious. Then medical experts developed treatments and vaccines, and the affliction disappeared, but not entirely. I recommend that if you can get hold of this, you need to read it. It's really interesting. So I'd just like to finish from this warning from WHO. So WHO warns that in the context of the COVID-19 pandemic, WHO reiterates to all countries globally that vaccination and epidemiological surveillance for vaccine-preventable diseases should be considered essential health services and should not be interrupted. Given the decline in diphtheria vaccine coverage in many countries, WHO also reminds member states that it is important to have a plan to vaccinate the most vulnerable populations and to maintain a permanent supply of diphtheria antitoxin to control potential outbreaks. And just to give you an idea, globally, more than 17 million children did not receive the first dose of DPT vaccine in 2020, as the coverage in fact fell to levels not seen in 14 years. So a real stark warning. Okay, I think that's it. And I'd like to just acknowledge, first of all, Mark Muscat, Mark for providing the diphtheria data slides, all the WHO regional officers for their constant collaborations, and of course, all the colleagues at NEQAS for the EQA diphtheria study. Thank you very much.
Right, okay, thanks a lot for that excellent presentation, very comprehensive, detailing clinical information, epidemiological information, and of course, laboratory aspects of diphtheria diagnosis. And I'm very happy that diphtheria has been put on this webinar because awareness is, of course, the first step towards having proper surveillance in each and every country. And we have a few questions actually relating to the microbiological surveillance. The first one coming from Afamia Jabour, where she asks that in countries with limited laboratory infrastructure, how to achieve the microbiological surveillance? And related to that, the second question is, are there any rapid tests or point of care tests in the pipeline to use such context? I noticed that you had already addressed this point, but perhaps you can elaborate a little bit more on the challenges that countries with limited laboratories, limited infrastructure of laboratories have in some countries.
Yeah, thank you. Thanks for the questions. Yeah, this is in fact, it is a major challenge, and we are, doing our very best, working with WHO to try and enhance lab capacity in these countries by holding workshops, supplying materials. That's another area that I didn't really discuss. We supply materials to specific areas. So we have very, if you like, very close collaborations with many countries within all the regions. The other question on the rapid tests, yeah, I mean, we had a wonderful test, a lateral flow test, very similar to the COVID LFTs. This is being revived at the moment via a Marie Curie EU fellowship that has been undertaken by one of our Russian colleagues, Dr. Slava Melnikov. And we're at the point of we're looking for a manufacturer that could produce these strips. It's a great test. We just need somebody to manufacture them. That's the whole point. I think they would be absolutely useful, essential, particularly in countries, in developing countries. Yeah.
Right. Yeah, that's a major challenge. We have another question from the same person. The difficulty with nasal and pharyngeal swabs is the growth of the tracts, the nasopharyngeal tracts, normal flora. Is there any really good selective medium to identify Corynebacterium?
Yeah, well, the primary isolation media, we use blood and tellurite. Many labs tend to use Tinsdale medium, which is highly selective. And I would certainly recommend it as part of the primary isolation panel, if you like. It's easy to make. The only disadvantage is it's not stable. Its stability is approximately, I would say, 3 weeks, 3 to 4 weeks. So it has to be made on a regular basis.
The last question we've got from the audience is from Declan Smith. And he asks, are phenotypical tests, for example, the ELIC, still used combined with PCR-based technologies?
Yes, always, because of the emergence of strains that may only possess the toxin gene, but may not necessarily express the toxin. We always confirm any PCR positives, or we strongly recommend confirming PCR positives with a phenotypic test, with the ELEC test. And this is normally done in the national reference labs within each country.
Great, thanks. We have no further questions, but I'd like to thank you, Andrella, for also stressing the importance of vaccination as the major tool to prevent diphtheria occurring, sporadic cases or outbreaks. That's the only tool we have to prevent diphtheria. Luckily, we have also included in the WHO immunization agenda the DTP3 coverage as a benchmark for monitoring progress of countries' immunization programs. And we always encourage countries to reach as high level of coverage as possible, both at national level and also at district levels. Because if the percentage of coverage goes below around 85% or so, then there's a risk of number of susceptible individuals accumulating and therefore the risk of outbreaks. And this is particularly important in places where people are living in overcrowded conditions. And therefore, this is very important, especially where refugees and migrants are gathering, especially when their vaccination status is not known. So thank you for bringing that up again on vaccination of diphtheria. I see there's another question coming up. Could you give any guidance to identify C. diphtheria only using blood agar for throat swabs?
Sorry, can you say?
The last question that just popped up now on the Q&A is, could you give any guidance to identify Corynebacterium diphtheria only using blood agar for throat swabs?
So not using any other media. I guess, I mean, if blood agar is the only medium available, if that's the question, then I would strongly recommend microscopy for any suspect colonies, followed by other individual tests. Where was that question coming from? Was it from a particular country or?
From a person, Nambaja Shirani Chandarasari.
Oh, okay.
That was Sri Lanka.
Ah, okay. Yeah. Well, there are single tests that we would recommend that are quite simple, which I described in my talk. So they could be used in combination with microscopy for presumptive identification. Again, all these methods and algorithms are explained fully in the manual. And as I said, I would be happy to send copies of the manual to anybody that is interested.
I've just sent the link, Androda, that was provided to us. But I just want to make everybody aware that this was not the very final version. The very last version, there were some edits that needed to be done, very insignificant edits. And therefore, I've asked WHO to upload the version that has been sent by email, to upload this new version through the link. So, I mean, the text is the same, it's just some minor editing, but that will probably be the same link for the very latest version. Yeah.